So this pathway is divided into different steps like initiation, nucleation, forming a pre-autophagosome, and further maturing of the pre-autophagosome into mature autophagosome, which further fuse with a lysosome to degrade its components. So the first step of initiation starts at ULK1. ULK1 is a serine-thiorein kinase which basically relays phosphorylations which are critical for autophagy. This is one of the most important steps where things begin and signals are sent towards going forward with the autophagic pathway. So ULK1 is phosphorylated by AMPK, which is AMP kinase, which can also inhibit mTOR-C, which eventually inhibit ULK1. So if mTOR-C is activated, then there will be no autophagy. So via signal from AMP kinase, when ULK1 is activated and phosphorylated, it binds to three different proteins and form a ULK1 complex. So ULK1 complex, once formed, eventually activates another protein via phosphorylation. It's called BECN1 or the Backlin-1 protein, which recruit these three other protein, the BP protein and ATG14L. ATG stand for autophagy related genes, and all these ATGs play a central role in making a mature autophagosome. So once this complex is formed, it's called the PI3K complex or phosphatidylinoisotol kinase complex. Once this complex is formed, it will phosphorylate a phospholipid called phosphatidylinocytol. This membrane comes from either ER or Golgi, or it could come from mitochondria. It depends in the cell. So once it binds to the membrane, it will phosphorylate this membrane at the 3' end. Once this step is complete, other proteins like ATG7, 12, and BP will be recruited onto this membrane. This step is called the pre-autophagosome, and it's also the step we call nucleation. It's a step where the autophagosome is conceived, or the first step where it comes into being. Further from here, after this, there are two complexes forming. Both of them act as an ubiquitination-like conjugation system. So the first one involves ATG4, 3, and 7, and the other one involves ATG16, 5, and 12. Both of these ubiquitination-like conjugation system will help to form a phagophore, which again means forming autophagosome, and they will recruit a protein called LC32. Now LC32 comes from here, codes the autophagosome, and it's a principal marker of whether autophagosome will form or it will not form. So ultimately, this happens at this stage. Then further, other proteins like P62, which is another ubiquitin cargo binding protein, it adds to the growing phagophore membrane and acts as a selective receptor for different substrates inside the cell. So just quickly going back to the LC3 and LC31, LC31 itself is a important marker for which aids in maturation and elongation of the phagophore via LC32, which is the lipidated form of LC31, which gets activated via ATG3-7 complex. So once that's done, further what happens is this phagophore will continue to grow along with the LC32 and makes a mature autophagosome. Once the autophagosome is completed, we have this double membrane structure, This is called a maturation stage of the autophagy. Finally, a fusion step will occur where mature autophagosome binds to a lysosome, and autophagosome will releases its content into the lysosome, and these proteins are degraded by the proteases inside the lysosome and is recycled back to the cytoplasm.